Good afternoon. Okay, students. So yesterday we were talking about antibody, right? So I showed you the picture of antibody, right? And we had a lot of discussion on the very basic aspects of the antibody molecule, right? And we also discussed how the structure of antibody was deduced, right? So today we will focus on the detailed structure of antibody, right? So अभी कल हमने जितना पढ़ा, so it was the very initial part, like कि कैसे with the use of some enzymes, how the structure of antibody was reduced, right? उस टाइम पे सिर्फ उतना ही पता था कि ये वाई शेप्ड होता है, राइट? और इसके डिफरेंट पाथ्स होते हैं, ओके, डिफरेंट चेन्स होती हैं, राइट? बट उसकी मॉलिक्यूलर स्टडीज तब तक नहीं हुई थी, राइट? सो by this time what you know about antibody molecule is the very basic part, right? And you know the names of few scientists who actually worked on this aspect, and they also got Nobel Prize for this work, right? So we talked about different domains of antibodies, right? So before I get into the details of these antibodies, let's start with the quiz. Okay, so I'm just going to assign the MCQs to you, and then we will discuss. And I'm sure if you score good today, then it means you have understood the basic concepts of antibody. Okay, but if you find it difficult, then maybe I'll have to take some more time to at least make you understand what we have discussed about antibodies till this time. Okay, so let me just share it with you. Okay, students, so I have assigned it to you. Is it visible at your end now?
Yes, sir,
Yes,
it is.
yes, I've got the notification. Okay. Uh, Read the questions carefully and then give the answer. Okay. So students, I can see 3 students have submitted. So now I think maybe in 2-3 minutes, all of you submit. So students have started submitting. So students, you can start submitting. Okay, so half of the class has submitted the response. Students, submit fast now. Students, start submitting now. Hello. Hani sir. Okay, students. So let's discuss the answers. Okay. Now the first question was, how many light chains are present in a molecule of antibody? Okay. So again, you just need to remember that structure. Okay. So there were total of 4 chains, right? 2 heavy and 2 light chains. So the correct answer is 2 light chain. Okay. And uh, only 88% of you have given the right answers. So you can analyze uh, here. Okay. Question 2 was, single light chain in antibody molecule consists of how many amino acids, right? And here only 70% of you have given the right answer. Okay. The correct answer is 220 amino acids, because if you remember, in a light chain there are 2 domains, right? And one domain is of around 110 amino acids. So in a light chain there are 2 domains. So 2 domains means 110 plus 110. So a total of 220 amino acids. Okay. Question 3 was, Rodney Potter used which reagent to reduce the structure of antibody molecule? Uh, Now, a lot of students have confused here. Only 63% of you have given the right answer. The correct answer is papain, not pepsin. Okay. So Potter used papain and Nisnov used pepsin. Okay, clear? Next question was, pepsin cleaves the antibody molecule at which point? So it's below the disulfide bond. Okay. So papain cleaves above the disulfide bond and pepsin cleaves below the disulfide bond. Okay. Question 5 was, Nobel Prize for reducing the structure of antibody was given to. So in this, all of you have given the right answer. It is Rodney Potter and Edelman. Okay. And question 6 was, FAB means. Okay. So again, for 82% of you have given the right answer. It's fragment antigen binding. Okay, it's not fragment antibody binding, because it itself is antibody, right? So it is a fragment which will bind to the antigen. So FAB stands for fragment antigen binding. Okay. So overall score is less than 10 out of 12. Okay. So overall you have made a good understanding of the structure of antibody. Okay. So let's continue with the slides. Let me share the slides. Okay, students, by the time it gets loaded, do you have any question? Okay, so this we have already discussed yesterday. Let me just come back to this point. Okay, because we just talked about the work of two scientists yesterday, and we have to talk about the third scientist today. So as I said, Potter, he used the enzyme papain, okay, and papain, it made a cut above the disulfide bond. So you can see this is the disulfide bond and the cut is above the disulfide bond, okay. And Nissanov, he used pepsin, okay, and this pepsin, it cuts below the disulfide bond, okay. Okay, so Edelman and Potter, they subjected antibody to mercaptoethanol. Okay, so these two scientists, Edelman and Potter, they used mercaptoethanol, and what mercaptoethanol does, it irreversibly cleaves the disulfide bonds. Okay, so if you put this antibody molecule in mercaptoethanol, it will break these disulfide bonds. Okay, now if these bonds are broken, okay, then the chains they get separate, right? So after breaking these bonds, they did chromatography, okay, and they observed heavy chain and light chain, okay. So with this, they know that there is heavy chain, there is light chain, there is FAB region, and there is FC region, right? So from all these experiments, the initial structure of antibody was deduced, okay. Right, so we have talked about the work of three scientists, and now we will discuss antibody in more details. Okay, so students, obviously you can understand when this structure was deduced, right? Scientists had very little techniques or technologies at their disposal, right? So obviously they could have done very limited work, but that work was still good enough, right, to make us understand about the structure of antibody. Okay. Okay, let me share the slides now. Okay, so now you know that what antibody regions are there. So one is your FAB region and the FC region, right? You have understood the importance of FAB region, right? AB is antigen binding. You know this, you have already answered it, okay? So FAB region, so this is your antibody, this is antigen. FAB region will go and bind to the antigen, right? But what is the role of this FC region? Okay, so let's understand this. So the FAB fragment can detect antigen. So we are talking about the upper portion. Okay. So this FAB region, it can detect antigen, right? It can precipitate the antigen. It can block the active site of toxins or pathogen associated molecules. It can block the interaction between host and pathogen associated molecules, right? So FAB region has its own work, right? To detect and bind and precipitate and block, right? So that is the work of FAB region. But this FAB region, it cannot activate inflammatory and effector functions associated with the cells. Okay, so it cannot activate inflammatory and effector functions of the complement system. Okay, so you can understand that uh, FAB region has its own work. F, uh, FC region has its own work, and that is very important because the effector functions have to be activated. The complement system has to be activated, right? Antigen processing has to be done. Okay, so for all these activities, and of course, we are going to talk about these effector functions. We are going to talk about the complement system. We are going to talk about the antigen processing pathways, right? So FC region has these very important roles, okay, which we are going to talk about later, right? So we'll have 3 classes on complement system, 3 classes on antigen processing presentations, right? Okay. So coming to the functions of different domains. So now you have just seen the different domains of antibody molecule, right? So if I just draw it, right, it is something like this, right? And it has different domains. Right? So these are the different domains we have already seen, like this is your uh, VL, right? And this is your VH. So this is one domain, right? And this is your CL, right? The constant region domain of the light chain. And this is your CH1. Okay, so the first domain of the constant region of the heavy chain. Okay, so each of these domain have different function. Okay, let's understand this. So obviously the first domain, the VH and the VL domain, their function is antigen binding. Okay, so the antigen will bind through these variable domains. Okay, let's see CH1, CH3. So this 1, 2, 3, 4, these constant region domains, they are the genetic markers of immunoglobulin, right? So this region, right, this region determines the class of the antibody. right? If it is IgG, IgA, IgE, so that you can identify from this region, okay? And I will make you clear how we do that, okay? So these are the genetic markers of immunoglobulin, okay? Now the Ch2 domain, this, and so this is your Ch2, this is your Ch3, right? So these Ch2 and Ch3 domain, they are the C1q binding site, okay? So again, don't worry about it. Um, Okay, so we are not going again, we are not going to talk about this. C1q is a complement protein, so इसको हम तब बात करेंगे जब हम complement system की बात करेंगे. But complement system activate होने के लिए some complement protein has to bind to this antibody. Okay, so it binds on these regions, right? So that is the importance of this domain. Okay, now Ch2, Ch3 domain in IgG, it is helping in binding to placenta, right? So I'm sure you know that some of the antibodies can cross the placenta, right? So, so as to provide protection to the fetus, okay? Again, if it has to bind to the placenta, it is through the Ch2 and Ch3 domains, okay? And again, one of the domain helps in binding to the receptor, okay? So let me just show you this. So you see this R is receptor, okay? Now you see this Fc, this is the this region Fc, right? And this you will understand later, but this is the class of antibody, right? So suppose this antibody has to bind to a receptor, okay? So how it will do this? Right, suppose on a cell there is a receptor, okay? So this antibody, right, and again, this side you have antigen, right, bound through the FAB region, and this FC region will bind to the cell through the FC region, right? So it is binding through this receptor, okay? So this binding has to occur, and it's again through the Ch3 domain. So you can understand each domain, okay, has a specific role to play in the functioning of antibody, okay? Okay, so okay, so I hope this is clear. Any question till this point? Is everything clear?
Yes, sir.
Okay.
Yes.
Okay, now what I want to tell you is अभी तक हमने जितना भी स्ट्रक्चर पढ़ा, राइट, वो बहुत बेसिक पढ़ा, राइट, बट विद द एडवेंट ऑफ टेक्निक्स, विद द एडवेंट ऑफ मोर टेक्नोलॉजीज, नाउ साइंटिस्ट कैन डू मोर डिटेल्ड स्टडी ऑफ any molecule, right? Not only antibody, but for that matter, any protein, any molecule, right? So, uh, before I actually come to the structure of antibody, there are few terms which I want to make you understand. So let me again share the slides. Okay. So now the scientist had this technique of sequencing this protein, right? So you can take the protein, you can do its sequencing, okay? You can get the nucleotide sequence, and you can get the peptide sequence, amino acid sequence, and then you can make out a lot of analysis, right? But if you have to do such studies in the lab, you need to have a large amount of the molecule of or the protein, right? अगर मैं तुम्हें सिर्फ एक मॉलिक्यूल दूंगा, it would be very difficult to work with that. ठीक है, वो सॉल्यूशन में एक मॉलिक्यूल मिलेगा ही नहीं. So you actually need to have a large amount of protein to do its study, right? So for doing sequencing studies, the scientist needed to produce a large amount of antibodies, right? So उसके लिए उन्होंने क्या टेक्निक यूज की वो मैं बताता हूँ, but before that just see this term which is multiple myeloma, okay? And what do you understand by multiple myeloma? So it's, I'm sure you have heard this term. Myeloma is cancer, right? So it's the cancer of antibody producing cells. Okay. So what is multiple myeloma? It's a cancer of antibody producing cells. And which cells produce antibody? It's the plasma cells. Okay. So in the patients of multiple myeloma, the plasma cells divide repeatedly in unregulated way, right? So what is cancer? In cancer, the cells, they divide repeatedly in unregulated way, right? So that is cancer. When we are talking about multiple myeloma, it is specific for the antibody producing cells or the plasma cells, right? So the plasma cells, they divide repeatedly in unregulated way in patients of multiple myeloma, right? Now these plasma cells, these cancerous plasma cells are called myeloma cell. Okay, so multiple myeloma is a disease condition, right? The plasma cells which are cancerous are called the myeloma cell. Okay, now in these myeloma cell, the protein synthesizing machinery and secretory functions are not altered. Okay, so ये जो माइलोमा सेल्स हैं, ये जो प्लाज्मा सेल्स हैं, इनकी जो प्रोटीन सिंथेसिस मशीनरी है, राइट, या जो इनकी सिक्रीटरी फंक्शंस हैं, that are not altered. So they will keep making the antibodies, they will keep secreting the antibodies, right? But the only thing is they are cancerous, okay? But जो उनका basic function है antibody produce करने का, that is not altered. So they will continue to secrete specific antibody, right? So myeloma cells continue to secrete specific antibodies, okay? And these specific antibodies are called myeloma protein, okay? So these are the antibodies produced by the myeloma cells, okay, in patients of multiple myeloma, okay? So are these three terms clear?
Yes, sir.
Yes, sir.
Sure. So one is multiple myeloma. It's a disease condition. It's a cancer of the antibody producing cell. Okay. These patients will have cancerous cells, which is your myeloma cells. Okay. So myeloma cells are the plasma cells, cancerous plasma cells, but their antibody producing ability is there. Okay. So they will keep secreting antibodies, and these antibodies are called the myeloma protein. Okay. Now, why I've used these terms, why I'm telling you about this is that if you have to carry out sequencing studies in of these immunoglobulins or antibodies, you have to create this in mice, right? And obviously, because uh, the plasma cell is secreting, it is multiplying, so there will be a large number of such cells, okay? And of course, they will keep producing the antibodies. So you can get a large amount of myeloma protein or that specific antibody. So if you have to use antibody for some sequencing studies, or you have to develop it on a large scale, then you can develop using this technique. Okay, clear? So in patients with multiple myeloma, myeloma proteins are up to 95% of total serum immunoglobulin, right? Just imagine, in patients में जो antibodies हैं, 95% are coming from the cancerous plasma cell. Okay, so you can see the impact of this cancer, right? Now, in these patients, they also secrete excessive light chains. Okay, and these light chains are there in the urine of the patients, right? And these light chains are called the Benzie-Jones proteins. Okay, so these are the scientists who discovered these proteins. Okay, now students, again very careful. Benzie-Jones proteins, if I talk about them, so they are only the light chains. So the excess of light chains which pass in the urine of a patient are called the Benzie-Jones proteins. Sometimes when we ask the students what are Benzie-Jones proteins, so they will say the antibodies in the urine of patient. So we are not talking about the whole of the antibody. We are just talking about the light chain which is there in the urine of the patient, not in the blood, right? So those are Benzie-Jones proteins, and multiple myeloma can arise spontaneously, okay? And it can also be induced by injecting mineral oil in mice, okay? So if you have to carry out some experiment in the mice, in the lab, you can just take mineral oil, inject it into the mice, and there will be multiple myeloma, okay? So with the help of this method, the scientists were now able to generate a large amount of antibodies, and that they can use for further studies. Okay. So students, I'll just stop here for attendance and then we will continue with this. Okay. So any question at this point coming to your mind?
Sir, too much of antibody, won't that be a risk?
Uh, yes, Adish, because the thing is, we actually don't need it, right? In our system, things are regulated. Immune system is regulated. It gets on when there is an infection, and then it comes back to its normal condition when there is no infection, right? Why to waste energy when there is no need, right? So yes, if there is excess of antibody circulating in the system, it will create pressure on other systems, right? So these antibodies have to be removed also. Okay, so it's really not a good situation, right? And because they are specific, so only a specific type of antibody is being produced, which is actually not required by the body. Okay, so there is no use of having that antibody in your system. So so it can, yes, excess of everything is bad, right? So it's not required, it will create problem. Okay.
Sir, yes.
Yeah.
Sir, and what usually does happen to those mice which have been injected with the mineral? I mean, do they get exhausted from producing too much of antibodies?
Can you repeat? You are not properly audible, Adish. थोड़ा जोर
Yes,
से
sir.
बोलो.
Yes, I was asking what usually happens to those mice? Do they usually get exhausted from producing too much of antibodies, or do they eventually die? Or
See, they will eventually die from cancer, right? So I'm saying eventually, right? So, but lab animals, obviously, there are a lot of ethical concerns, right? You have to get ethical approvals for this. You have to induce cancer in them. you have to take out the antibodies, and then in most of the cases either we kill those animals, right, or they will definitely die their natural death because of the disease, right? So we are producing them for this purpose, and you will see a lot of animal models, like I will be talking about them, right? So ultimately, obviously, those lab animals, and you cannot just let them go, right? Suppose you have completed your experiment, your work is done, you cannot let them go because you have made a lot of changes in them, right? And so suppose you have made some genetic modification in a mice, and then you are allowing it to go out and breed with the normal population, so that could be a risk, right? So uh, so you actually don't allow those mice to get out from the lab, so they are eventually killed in the lab, right?
Sir, do we also need ethical permission to conduct tests on lab?
Yes, so not only for animals. So see, when you are doing research, there are a lot of protocols. Okay, if I have to take blood sample from a patient, okay, or even from a normal healthy individual, I will have to get ethical approval. Okay, so in all the universities, in all the hospitals, in all such organizations involved with research, right, you have to first get the ethical approval. Okay, so what happens? Suppose I have made a project, okay, and I say that I need to collect 10 ml of blood sample from the patient. Okay, so there is a committee, and it's a big committee. It includes some externals, right? ऐसा नहीं है कि organization के चार लोग बैठे और ethical approval दे दिया, right? So there are people from different fields. उसमें एक law का भी बंदा बैठा होगा, उसमें एक humanities का बैठा होगा, उसमें science का होगा, उसमें so they all will try to understand why you require blood, okay, what are the risks associated with it, right? Are you going to give any money to the volunteers, right? Because when you are doing such studies, you cannot pay to the volunteers, okay? Again, it is not ethically right, right? So all these factors are analyzed, assessed, whether I am going to give unnecessary problem to the patient, okay? Whether I am taking the blood one time, 10 times, 5 times, and if I am taking 5 times, I have to justify why I am taking 5 times, right? If I am taking 10 ml, I have to justify why I am taking 10 ml, right? अब एक बार I remember एक student था, he asked for 10 ml of blood of infants, right? So the ethical committee वो सारा discuss करने के बाद they said no, you cannot take 10 ml from an infant, 6 महीने से छोटे बच्चे का मैक्सिमम तुम्हें 1 ml मिलेगा, एंड यू हैव टू लाइक think about it, जो भी है, right? So first thing is to get the ethical approval. It's not easy. Another thing is they will also ask, apart from your research work, right, they will also ask for the consent form, okay? That is again a very interesting thing. So you have to prepare a consent form. What is your research, why you are doing it, what could be the possible harm of participating in that particular study, right? So there are a lot of things which are mentioned on the consent form, even the number of the person who is taking the blood, okay, whom you can contact if there is any problem, okay? Another very important thing, suppose मैं गांव में गया, राइट, और मैं एक इंग्लिश का कंसेंट फॉर्म लेके चला गया, सो डू यू थिंक the person will be able to read that consent form. उसमें क्या लिखा है उसको समझ आएगा नहीं. Okay, so I have to make a consent form in their language, right? And I face this difficulty. So you know about my Indo-US project, right? So obviously when I made that consent form, it was in English. Okay, and the US also, that organization also approved the English version of this consent form. But the problem was I have to go to uh, the community where very like poor people are there, right? So they cannot understand the English form. Okay, so I had to make another consent form, so I translated that into Hindi, and then I used that form. Okay, and suppose when I remember when I was doing research in Punjab region, so I had to make the consent form in Punjabi, right? And then you have to take the signature from that person before you collect the blood. Okay, so there are a lot of things. So research is not that easy कि गए blood लिया और आके testing शुरू कर दी. Okay, so you have to follow a lot of guidelines and protocols. Okay, okay, students, let's have attendance, and if then time will be remaining, we'll have more questions. Okay, so Adish,
Present, sir.
Atoiba,
Present, sir.
okay, Ayushi,
Present, sir.
Sam,
Present, sir.
Devanshi,
Present, sir.
Ishita,
Present, sir.
Janvi,
Present, sir.
Manaswani,
Present, sir.
Namrita, Namrita,
Present, sir. Present, sir.
Nehal,
Present, sir.
Rupal,
Present, sir.
Sahiba,
Present, sir.
Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
okay, Tanvi,
Present, sir.
Tridev,
Sir.
Vivek, Yukta,
Present, sir.
okay, Yuvraj is also present. Vivek, are you there? Okay, okay. Okay, students, so any other question?
Yes, sir.
Yes, Tanvi?
So, sir, as you mentioned in your last slide that there are light chains in the urine sample.
Hmm.
So, I want to ask that why there are only light chains in the urine sample and not the heavy chains?
I don't know the exact reason, but maybe the light chains they are filtered through the kidney, right? But the heavy chains they are not filtered through it. I think this is the reason for it. But I am not sure. I'm not sure for this, okay? Yeah. Okay, so students, the time is up, so I'll have to end this class. And so now you know that the scientists now have this technique where they can generate a large amount of antibody molecules. Okay, so in the next class we will see how by using these antibodies they will uh, like reduce the details of the antibody structure. Okay, so this we are going to discuss in the next class. Okay, so till that time, keep reading, and if you have more questions, we can have a discussion in the next class. Okay.